If you ever remember Jimmy Neutron, the TV show from a long time ago on Cartoon Network or Nickelodeon, I remember there was one episode where they were fighting these viruses that were on the macro level. I thought it was really cool stuff. And so what I'd do in this video is take you guys through the virus as well as some subviral particles that are very important to understand if you're interested in medicine or biology. And so to get started, I've drawn to the left here a very basic virus that hopefully we've seen before. And we know that viruses are composed of 4 parts. And so the first part is this outer coat referred to as the capsid. The capsid is composed of proteins and lipids, and it is essentially just a way of containing the genetic material inside. I'll just refer to it as genes. And these genes, to keep this a generic example, can be either DNA or RNA. They can be double-stranded or single-stranded. So we've got 4 different types of genetic material we can have inside of our capsid. We've also got these things referred to as tail fibers that are on the end of the virus, as well as these things which are referred to as tail sheaths. And what will happen is, if we imagine we had some kind of cell here at the surface, there's going to be some kind of cell receptor. I'll just draw a block. And this receptor may or may not have some affinity for this tail fiber. And so if the tail fiber has an affinity for the receptor, the virus will bind onto the surface of the bacteria or cell. And so you'd get these little viruses attached on the surface. And what happens next is, because there are different types of bacteria, viruses, what will happen is we may or may not see something referred to as enveloping occur. And enveloping means that the viral capsid fuses with the membrane. And so in that case, all of this stuff would become part of the surrounding phospholipid bilayer, but this isn't always the case. And so sometimes if we have non-enveloping viruses, the genetic material will be injected into the cell through the tail sheath, and the capsid, the empty capsid, and the rest of the components of the virus will just remain on the outside. And so after the virus infects the cell, which is another way of saying the virus tail fibers have bound to the receptors, and the genes have been inserted into the host cell, the next thing that's going to happen is, depending on what type of genetic material the virus has, we will now induce translation. And so if the genes are DNA, then they go to the nucleus. And in the nucleus, they'll be converted into mRNA. If it's a eukaryotic cell, it may require some processing to derive a mature mRNA before we can actually translate the genes into proteins. If we have something referred to as plus sense RNA, so if the genetic material of the virus is RNA and it is referred to as plus sense, the plus sense RNA can be directly translated. So you can think of it as mature mRNA. But if we're dealing with minus sense RNA, we will need to have a copy of the mRNA or the RNA to have a complementary strand. And this complementary strand can then be translated. So it requires an additional step, which, if you're looking at things from an evolutionary perspective, additional steps might add room for problems. So plus sense RNA would probably be more common to find than minus sense RNA types of viruses. And then finally, there are viruses referred to as retroviruses. And if we have a retrovirus, these are particularly nasty because the DNA, so it will, the retrovirus will have RNA as well as a mRNA reverse transcriptase. And what happens is the virus will contain the genes to create a reverse transcriptase that will turn its RNA into DNA. And once the DNA is created, that DNA will be incorporated into the host's genome. And this is a big problem in patients with HIV, for instance, because once the host genome has been infected and the viral DNA, once RNA becomes incorporated into it, the host cell will be infected indefinitely. And so the HIV is caused by a retrovirus that is particularly nasty to deal with. And so what happens next after the virus has bound to the cell, injected its genome or genes, and the genes have been translated into proteins to create the progeny. The next step is that somehow the progeny will need to be exported from the cell to go infect more cells. And so the way that we're going to do that is two ways. We can either do a pseudo apoptosis, in which we trigger cell death, and once the cell dies, the membrane will become permeable and the progeny will be able to become exposed to their environment and infect more cells. Or we can perform something referred to as cell lysis. And what happens in cell lysis is the virus will create so many copies of itself that eventually the cell will just burst due to physical reasons. And that is another mechanism by which we can get the viruses out to infect more cells. And from a virus perspective, this second method is not that great because you just killed your host and you can't make any more copies of yourself from that same cell. And so there are types of viruses that are able to not do this. And there's a pathway referred to as the lysogenic path. And viruses that do this, it's referred to as the cycle actually. When viruses do this, they become part of the host's genome and they are able to survive indefinitely alongside the host. But the problem with this is it is a metastable relationship because what will happen is stimuli can cause the lysogenic virus, which is sometimes referred to as a provirus, to become lytic. And when a virus is lytic, it is in the rape and pillage mode, meaning that it will go about and use all the cell's machinery at maximum capacity without regard to the host, and this will cause cell death. And so that is a big problem. And then finally, to wrap things up, there is something referred to as prions. So these small or subviral particles, and these are pathogentic because they're essentially evil protein chaperones. And so, as we'll call in the protein folding process, as the amino acids are forming three-dimensional structures, secondary and tertiary structures, you need to have certain proteins helping in this process because these are very complex structures, and on their own they would take enormous amounts of time to maybe not even happen. And so what these prions are they're very small segments of proteins that will interfere with the protein folding process. And so if a host cell has prions present that interfere with the manufacture of some kind of critical protein for the cell's survival, it can cause cell death and therefore be toxic. And so this wraps up an introduction to the virus and subviral particles. I hope you guys find it useful. Let me know if you have any questions, and thanks for watching.